Hello, my name is Janet E., and my presentation today will be based on the journal article titled Curvature Recognition and Force Generation in Phagocytosis. Phagocytosis is the ingestion of a smaller cell or cell fragment, a microorganism, or foreign particles by the infolding and closure of the cell's membrane, what we learned as cellular eating. We briefly talked about phagocytosis in the beginning of the semester, and it is explained in more detail in the Biology 6 class. You may think that in phagocytosis, the phagocyte only forms a circle around the particles and takes it into the cell for digestion. But that's the easy way of looking at it. We will actually see in the presentation that the cell works pretty hard in attempting to bring the particle in. The two key players in this experiment were the dictyostelium cells, shown on the left, and the Saccharomyces cerevisiae cells, shown on the right. Dictyostelium is a genus, and these guys are our amoebas, and these are the ones that will be doing the eating, so they are the phagocytes. The Saccharomyces cerevisiae are the budded yeast cells, and these guys will be the ones who are eaten by the phagocyte. The experiment will show that the phagocyte has three options. The first option is that the cup extension can cease at the constricted region, and the constricted region is essentially the region where the mother cell is connected to the bud cell. So the cup extension can stop there and attempt to sever the particle there, and thus only engulfing the section that was surrounded initially by the phagocyte. The second option is the extension past the constricted region until the whole particle is engulfed. The third option, if both previous options are unsuccessful, is that the entire particle will be released. So unfortunately, therefore, the phagocyte will have nothing to consume. To monitor phagocytosis in these guys, green fluorescent protein was fused to another protein called IBAR-A. Green fluorescent protein is a protein that is used by many researchers to see when a protein is made and where it goes. And this is done by joining the green fluorescent protein gene to the gene of the protein of interest, so that when the protein of interest is made, it will have the green fluorescent protein hanging off of it, so it'll fluoresce a nice green color. The IBAR-A protein is essentially a protein that regulates actin dynamics, and remember that actin is a protein that is responsible for shape changes, cell locomotion, and muscle contraction. And this is important in phagocytosis because the phagocytic cup does contract and regress to enclose the particle it's seeking to ingest. So there's a lot of actin involved in phagocytosis. And this is a figure here. This is a time series showing the actin in green with the cell interacting with two budded yeast cells. One budded yeast cell is on the right, and the second one is located at the left side of the phagocyte. The phagocyte first encounters the budded yeast cell in the first row, and the phagocyte encounters the second budded yeast cell on the second row. The last row is simply the interaction with the original budded yeast cell, and in the last slide it's actually the severance of the mother cell from the bud cell. And this is actually a video of that figure. So the arrows point to the budded yeast cells, and again it first interacts with the budded yeast cell on the right. So there's actually a strong accumulation of actin at the neck or the constricted region of the budded yeast cell. You see it linger there for a bit because it's kind of determining whether it should engulf the remaining particle, if it should sever it there, or if it should release it. So it looks like it decides it's going to release the particle and attempt to engulf the particle to the left. So again, you see the strong accumulation on the left budded yeast cell, and that attempt is also released. Finally, it does go back to the original budded yeast cell, and it decides to just go ahead and sever it there. And you can actually see that the mother cell is separated from the bud cell. Phagocytosis is the ingestion of a smaller cell or cell fragment, a microorganism, or foreign particles by the infolding and closure of the cell's membrane, what we learned as cellular eating. We briefly talked about phagocytosis in the beginning of the semester, and it is explained in more detail in the Biology 6 class. You may think that in phagocytosis, the phagocyte only forms a circle around the particles and takes it into the cell for digestion. But that's the easy way of looking at it. We will actually see in the presentation that the cell works pretty hard in attempting to bring the particle in. This is the last video that I will be showing you. This video will be slightly different from the previous videos. The previous videos, if you recall, is basically the severance of the mother cell from the bud cell and the release of the phagocyte from the budded yeast cell due to its incapacity to uptake the particle. So this video is actually going to reflect the uptake of the entire particle, as you will soon see. So there's that red fluorescent protein, which is the actin. It's not so strong at the neck region because it's actually able to go ahead and proceed with the uptake of even the bud cell. So it is successful at engulfing the entire particle, as you can see. So it does bring in the entire particle into the cell. And this is just a darker image of what was previously seen. So this concludes my presentation, and I hope you guys enjoyed it somewhat. One thing that I hope you get out of this presentation is that phagocytes are not unidirectional. They have several options to choose from when encountering a particle, so we cannot automatically assume that because it's a phagocyte that it will generally engulf anything it encounters.